My name is Grocott. I'm the chair of the Department of Anesthesiology and Pain Medicine here at the University of Washington. And it is my true honor to welcome our inaugural Tom Hornbein lecturer, Dr. Mike Grocott, who is professor of anesthesia and critical care medicine at the University of Southampton and the director of the National Institute for Health and Care, not healthcare, health and care, Southampton Biomedical Research Center. Dr. Grocott's pioneering work in perioperative medicine, extreme physiology, as well as human performance at high altitude, places him amongst the leading clinician scientists of our specialty. With the help of many in the audience, both online as well as here in person, and that includes friends, that includes colleagues, former trainees, as well as the family of Tom Hornbein, we are able to create this endowment, which I'm so pleased with, that we can now honor Dr. Hornbein with, and I read the official title, the Thomas Hornbein Endowed Memorial Fund. This fund actually is dedicated to the educational opportunities of our residents, and we are glad for them to have an opportunity tomorrow to have a fireside chat reserved with Dr. Grocott, as well as tomorrow's Grand Rounds lecture. This really is an opportunity to celebrate Dr. Hornbein, his legacy and his life. He was a former chair of the department, a legendary mountaineer, and certainly one of, I think you would agree, true icons of American climbing. Dr. Hornbein was celebrated not only for his historic climb in 1963, where he ascended the Mount Everest on the West Ridge by a new approach with his comrade Willie Unsold, a feat that redefined what's actually possible up in high altitude, but also for the extraordinary humanity, his intellect, his quiet courage, and his curiosity that shaped his more than 90 years. I can tell you from personal experience, I've met him 3 times, and then I've met him several times online, which was in the years up leading to his death, where he was still interested when I became first interim chair the second time and permanent chair. He was interested to hear about the progression of the department, new developments, how the residency's doing. And it was remarkable to see his interest, and that was in his early 90s. His life embodied a rare blend of scientific curiosity, as I said, but also fearless exploration, inspiring, I think, generations of climbers, physicians, and adventurers. It is especially fitting, and I think we couldn't have found a better speaker than Dr. Grocott, whose own research and expedition continue to push the boundaries of human performance in extreme environments, and for him to deliver this first lecture in Dr. Hornbein's honor. Thank you all for your generosity. We left a little paperwork alongside the aisles for you to consider or take with you and share with your colleagues and friends. With that, I would like to ask everyone to join us in welcoming Dr. Grocott to give this first lecture. Thank you for coming. Please silence your phones.
So Tom Hornbein, charismatic, kind, caring, incredibly talented, inspirational, definitely mischievous, an extraordinary individual, and I was fortunate enough to know moderately well, certainly not well. I suspect there are many people in the audience who knew him much better, but he was an inspiration, and I honestly cannot think of another named lecture that I would be more honored or happy to give. So it's a real delight. I'm very grateful that you asked me to come and do this, and it's a real delight to be here in Seattle in his home department, and to have an opportunity to talk a little bit about Tom. I've somehow jumped over there we go, conflicts of interest. Just none of them has anything to do with this talk at all, but just in case you were interested, conflicts of interest. And I'm going to focus principally on the climbing side of Tom. I suspect many of you will know and understand his role as an academic leader here and as a scientist and anesthesiologist, but I'm going to focus more on his climbing achievements, which I think, I mean, Grocott's touched upon, but it is hard to overestimate the magnitude of what he achieved on Mount Everest. So I'm sure you all know Mount Everest, and don't worry about the detail on this slide. It'll become clear in a moment. This is a list of the first 15 people to climb Mount Everest. So the red box at the moment is around Hillary and Tenzing. They climbed on May 29th, 1953. It was a very rare event that climb was repeated in the first few years. So 1956, the Swiss came along. 4 of them got to the summit a little bit earlier in May. A few years later in 1960, it was controversial at the time. In fact, a lot of people didn't believe that this climb had taken place, but I think there's good evidence now that it did. The Chinese reached the summit from the other side by a second route. So Hillary and Tenzing had gone up the southeast ridge, which I suspect meant you will have heard accounts of Everest. You'll have read about it. You'll have seen on the television or in films. So up from the Nepali side, up through the Western Cwm, Lhotse face, the southeast ridge, which is the same route as the Swiss. The Chinese approached from the other side, so the northeast ridge. And then along came the Americans in 1963. 2 separate ascents. So Jim Whittaker with Nguyen Gombe Sherpa, making sure that they kind of planted the flag. So they went up the southeast ridge route and successfully climbed very early in May. And then they also had this extraordinary idea of trying to climb the West Ridge. So nobody had any idea if it was possible to do that. And Tom and Willie were the 2 that really believed that this was going to be something that was worth doing. It was a challenge worth taking on, even though they knew the likelihood of success was relatively low. And so they set off. It's not the highest quality picture, but it's one of the better illustrations of how this works. So south side of Everest, you can see Lhotse in the center there, Everest towards the left, and the sequence of camps going up towards the summit in the dotted line, which is the standard southeast ridge route. And what William Salt and Tom Hornbein did was follow the continuous line. So they looped slightly back on themselves onto the West Ridge. I'll show you a couple of pictures in a moment. And then they went progressively up the West Ridge until it became essentially impossible to continue. So they didn't stop. They traversed out onto the north face of Everest and climbed a couloir that became known as the Hornbein Couloir, making Tom one of only 2 people ever to have a feature on Everest named after him. So the Hornbein Couloir and obviously the Hillary Step. And they continued up and they finally summited Everest late in the day and then descended by what is typically known as the normal route, so the Southeast Ridge route, becoming the first people to traverse the mountain. So brand new route, unknown if it was achievable, and the first and 2 of very, very, very few people. There's a handful of people who've gone up Everest one way and come down another way. Here's a picture of Tom, as I suspect very few in the audience will remember him in his younger days. This is on the 1963 expedition. And he's just, the middle picture is really to give you a sense of scale for Everest. So Tom and Willie are just tucked away in the bottom there, if you can see it, on the snowy ridge, which goes for quite some distance before you start to get to the rocky bit at the top. And then this is a picture of one of their higher camps. And this is focusing in, this is the Hornbein Couloir. So I can't move because of the microphone, so I'll gesticulate a little bit. But so the shadow towards the right of this image, you can see a snowy couloir ascending, going slightly to the left, slightly to the right, slightly to the left. And that's the Hornbein Couloir, which they successfully ascended, finally making it out to the summit. And I first when they got to the summit, they then, when they descended the other side, they met two others who had come up the other way. So Barry Bishop and Luke Jester, who had come up from the Southeast Ridge route, they were far too late. So they ended up then having the highest bivouac that had ever been done. And still to this day, very few bivouacs have been higher. And they finally descended down, and Tom was the only one who managed to reach the South Col without frostbite and managed to keep all of his digits. I first encountered this story when I was relatively young. So in '98, I spent 3 months climbing in Nepal. And when I got back to Kathmandu at the end of May, acquired all the climbing books I could in the shops. And those who've been to Kathmandu will know there's a lot of really smart bookshops. So all the climbing books I could. So I got hold of this copy of Everest, the West Ridge. And this isn't an original version, although it's got the same cover image. This is the Indian edition. And then was really fortunate a couple of years later, 3 years later, to meet Tom and get him to sign it. So this is my signed version of Everest, the West Ridge. Now, it turned out when I started doing this talk, I thought I had one signed version of Everest, the West Ridge, but it turns out that wasn't true. The next, I bumped into Tom at the International Hypoxia Symposia every 2 years in Lake Louise, occasionally in other places. And the next time he was incredibly generous to me was when we wrote this article. So this was, extraordinary case series of 4 that got us into the New England Journal of Medicine, but I guess it was a relatively unusual context. The story of publishing this was quite long and drawn out. We couldn't get it into quite a few journals. And then one of our colleagues, a chap called Mervyn Singer in London, said, well, why don't you just go for the New England? They'll like it. So we did, and then they pushed back and said, you've got to write a letter. And then we got help from all sorts of people. Amongst one of the most helpful was Tom. So he helped us refine this manuscript and get it through the New England review process. And so this is the end of the manuscript, and he is there credited. There you go. All our volunteers, all various other people, and Tom Hornbein, Eric Swenson, another local boy, not anymore, but was here for many years, and a couple of others who helped us. I then met Tom again. He did the 50th anniversary lecture at the International Hypoxia Symposium, and he had a brand new book at that point, which John Krakauer was involved with. So I was fortunate enough to have another signed version from Tom, also signed by John Krakauer with a somewhat less flamboyant signature. And in 2017, we had our 10th anniversary of our expedition. We were quite a long way behind, obviously. And we invited Tom, amongst others, to come over and lecture. We had a big 2-day meeting in London. And here's Tom with Jim Millage, for those of you famous high-altitude physician and physiologist from the UK, John Severinghouse, who also came over, Martin Feelish, who's a nitric oxide biology colleague of mine. And I'm so honoured to have a picture in which Tom and I feature in the same image. On the occasion of which it just happened to be this was my 51st birthday, which happens to be Tom's 54th anniversary. So I got my 3rd signed book with a somewhat longer message, which is the one probably I treasure the most. And I think it's probably hard, if you're not a climber, it's hard to put into context how impressive the feat of climbing the West Ridge for the first time, completely unknown, challenging, then descending the Southeast Ridge, doing the first traverse of the mountain. It's an extraordinary achievement. And I think the single measure that I found that probably reflects the magnitude of that achievement the most was this. So the American Alpine Club has only ever presented their gold medal 7 times. Twice, Hornbein managed to be the recipient. Once as a group member on the 50th anniversary, and then once subsequently in 2019. I think that gives you the measure of the magnitude of that climbing achievement. I'm going to go back to Everest now and talk a little bit about of Everest history, and then a little bit about the research that we've done on Everest. So Everest is not the tallest one there. That's actually the end of the ridge that comes round from Lhotse and Nuptse. Everest is the rocky peak in the center. There were a number of attempts to climb it, as you're probably aware, through the 1920s and then early 1930s, which were unsuccessful, but actually came remarkably close to success. So with and without supplemental oxygen, a couple of individuals got within about 500 meters of the summit, but they didn't quite make it. Everything stopped during the war. And after the war, the Chinese invaded Tibet, the north side of the mountain closed down, and all the approaches started to be from the south. The Swiss went in 1952 and got very close to the top. But I think, as everybody knows, the first successful summit was Edmund Hillary and Tenzing Norgay in late May 1953, just before the coronation of our now departed Queen. And the news came back, and it's sort of folklore that it's true that the announcement was on Coronation Day that this great British success of a British expedition. And I think everybody glossed over the fact that it was actually a Kiwi and a Nepali who'd successfully summited. It was a British-led expedition. What Hillary and Tenzing showed us was that it was possible for humans to reach the summit of Everest. What it didn't show us was whether it was possible to do that without additional oxygen. So they used supplemental oxygen. One of the reasons they succeeded, there are a variety of reasons, but one of the reasons is they had a new oxygen system was more efficient than those that had gone before. And if you read Hillary's account, he describes taking his oxygen mask off and feeling pretty rotten. And his view at that time was that it would not be possible for humans to climb Everest without additional oxygen. And that indeed remained true for about 25 years until these two gentlemen came along, so Reinhold Messner and Peter Habeler, two German-speaking Italians from the Tyrol who successfully climbed Everest without supplemental oxygen in 1978. There's a lovely side story to this in that they got a lot of criticism because I suspect, as most of us would think prudently, they took some oxygen with them just in case. So they left some stashed on the South Col. And because of the nature of climbing ethics, people felt that was like cheating. So Messner went back in 2 years later, in 1980, and climbed on his own by an entirely new route on the North Face without any oxygen anywhere near him, and successfully summited again to kind of put that one to bed. Hornbein and Unsold were the 14th and 15th to climb Everest. Messner and Habeler the 64th and 65th. When I went in 2007, we were just over 2,500. There's now more than 7,500 people have climbed Everest. So the rate of ascents is going up and up and up. I suspect this is very familiar to this audience, but just to give you some hooks to hang the hypoxia story on. This is a graph of barometric pressure horizontally versus altitude vertically. I apologize, this is in kilopascals. I'm from the other side of the Atlantic, so that's the way we work. And you can see in the center, there's a red triangle which says highest human habitation. That's pretty much where Everest Base Camp is in terms of altitude. Just around 5,000 meters. Everest Base Camp's about 5,300. And the oxygen level there, barometric pressure there's about half. Oxygen, the proportion of oxygen in the air remains the same. So the partial pressure of oxygen is about half what is at sea level. And the summit of Everest, 8,848 meters, just over 29,000 feet. The barometric pressure and the partial pressure of oxygen is about a third. So when we're going through some of the data we had, you keep those figures in mind, about a half at base camp and about a third when you get to the summit. And if you acutely expose humans to these kind of levels of hypoxia, certainly the summit, it's really bad for them. And the classic scenario is your James Bond movie, somebody shoots in the aeroplane, acute cabin decompression, and from essentially about 2,500 meters equivalent, you go up to about 8,500, 9,000 meters equivalent. And that leads to rapid loss of consciousness, and actually due to a paradoxical phenomenon of hypoxic hypoventilation, death within about 5 minutes. So loss of consciousness within probably about 60 to 90 seconds and death within about 5 minutes. So if there's one take home from this lecture, when the masks drop down, grab hold of them quickly and hope the pilot descends. Now, as I've told you, people can climb Everest without additional oxygen. And yet, if you acutely expose them, it's really bad for them. So what happens in between? And that I'm sure you know, is the process of adaptation or acclimatization that enables the body to tolerate low oxygen levels. And it's a very well described phenomenon. This is a graphic from a review in the British Medical Journal towards the end of the 1990s. And it describes all the different physiological responses that take place. And they all serve rationally, I guess, to increase oxygen transport from the atmosphere down towards the cells, which ultimately metabolize it. So you breathe a bit more, get a bit more oxygen to your alveolus. You have a greater hemoglobin, so you can carry more in the blood. The cardiac output increases, so you can pump that. You increase your oxygen delivery. The capillary density in the tissues improve, so the diffusion distance go down. So all great. Superb description of what happens at altitude, but not really an effective explanation, because if you take any one of these individual variables, they don't predict who does well at altitude and who does badly. Or if you like, sum them all together, so you take a measure like VO2 max, the ability of an individual to transport oxygen from the atmosphere down to their cells and to metabolize it, that doesn't predict how well someone performs at altitude either. Very counterintuitively. So when we went to Everest in 2007, this is one of a number of expeditions we've done to Nepal, but this is the one where we went to the top. We had two very clear aims, and that was partly to understand more about critical illness. So to use altitude, where a single variable changing, i.e. oxygen tension, could be controlled as a means of understanding hypoxia and critical illness. And secondly, to look at this paradox of the lack of explanation of oxygen delivery as to how well people adapt, and look for other explanations of the inter-individual differences in performance that you see. So we started from the presumption that it's really hard to tease out the effect of an individual variable like low oxygen levels in critically ill patients, because there's so much else going on. So patients come in with different conditions. We then throw different treatments at them. Their natural history varies over time, and the effect of an individual difference, like higher or lower oxygen, is very hard to tease out. If, on the other hand, you take relatively healthy people, you give them a very controlled ascent, and we matched the ascent of all our volunteers, over 200 of them went to base camp, you can start to look at inter-individual differences in the ability to adapt to lower oxygen levels. Why did we believe there might be some usefulness in this approach? Well, partly there was some genetic data available. This is from my colleague Hugh Montgomery, who I worked with for many years at UCLH. There was some genetic data available suggesting that the same genetic determinants of performance at high altitude also underpinned tolerance to hypoxia clinically. So this is two alleles of the angiotensin converting enzyme gene, the so-called insertional deletion alleles. And in the normal population, there's a perfectly balanced Hardy-Weinberg distribution. So 25% for each of the two heterozygotes, 50% for each of the two homozygotes, 50% for the heterozygote. And yet in the blue here on the left, you can see there's an overrepresentation of the I allele in individuals who successfully climb to high altitude. So this group happened to be British mountaineers who'd repeatedly ascended over 7,000 meters. Very, very few of them are DD homozygotes, having two versions of the D allele. If, on the other hand, you look at adult respiratory or acute respiratory distress syndrome, which is our archetypal illness of low oxygen levels in critical care, there is a survival benefit for having the same alleles, so the I allele, as seem to confer benefit in terms of performance at high altitude. So survival in the II group, just over 10%, mortality just over 10%, versus more than 50% in the DD group. And this has been replicated by other groups as well. So performance at altitude is not directly related to how much oxygen you can shift around the body. Now, I know this is not necessarily what we're taught in Physiology 101, but the data is compelling. And if we start by taking the example of Mesmer and Haberle, so you would expect perfect oxygen transport machines, given that they're the first individuals to successfully ascend Everest without additional oxygen. They were studied in great detail throughout the late 80s and 90s. Throughout the late 70s, 80s, and 90s. And essentially, the most remarkable thing about them was how unremarkable they were. This is one example. So all sorts of things were looked at: hemoglobin content, hypoxic ventilator response, all sorts of different physiological variables. This is VO2 max. The red box encompasses Mesmer, Haberle, and three other elite European mountaineers. The group that aren't boxed are some elite Sherpa mountaineers, and the green box is a set of Swiss couch potato controls. So Mesmer and Haberle were not that extraordinarily fit. Nothing about their physiology stood out as different from controls. Moreover, and this is some of our data from 2007, if you look at oxygen content in individuals who are acclimatized, so they've had sufficient time to increase their hemoglobin due to the low oxygen levels, to counteract the effect of their lower oxygen saturations due to the lower oxygen levels. In fact, the oxygen content is remarkably well maintained up to pretty high altitude. So don't worry about all the details of all the bars, but the darkest bar here is oxygen content. At sea level, if we go from left to right, Everest Base Camp at 5,300 meters, Camp 2 at 6,004, Camp 3 at 7,1, and then very high on the southeast ridge. And you can see that right up to over 7,000 meters in well acclimatized individuals, oxygen content is either normal or above normal. So any decrement in performance is not explained by a low level of oxygen in the blood. Something else is going on. It's not explained, and the measurements aren't here, but I can tell you that for any given workload, the cardiac output's for the same. So the anesthesiologist thinking through the oxygen delivery equation, the oxygen delivery is the same or greater than at sea level, and yet performance is decreased. So we set out to look at, in particular, what you might call tissue factors. So the function of the microcirculation and the function of metabolizing metabolism within the cells, and particularly mitochondrial metabolism. So essentially, it's not about the oxygen that's being delivered in the cardiovascular system as a result of respiratory effort. It's principally determined by the function of the tissues, the microcirculation, and mitochondrial function. And in order to do this, we had to do quite a lot of preparation. So this was taking all the equipment that we needed to take to Everest. We needed to be sure the investment of time and energy and other people's money that were required to run this sort of expedition. We needed to be very confident that the devices we were taking will both function and be reliable and measure accurately. So this is on the far side, you can see a chamber which can lower pressure down to the equivalent of about 9,000 meters. So all our kit was taken to hyperbaric, and you can see somebody with an oxygen mask on just peeking over in the background as we use a metabolic calibrator for the cardiopulmonary exercise testing, for example. And then we did look at more complicated structures, but this is a butcher's wagon that we parked outside our lab in North London that would go down to -20. And we again tested all the equipment for both reliability and calibration in that. So we knew that within the conditions that we were likely to expect near the summit of Everest, we would be able to measure reliably and effectively. We also tested the people. So anybody who was going to qualify to be on the summit team and a high altitude researcher on Everest had to successfully ascend an 8,000 meter peak before we went to Everest. So this is Choyu. It's the 6th highest mountain in the world. It's about 10 miles from Everest on the Nepal-Tibet border. It's 8,206 meters, so not a long way off short of Everest. And we put 16 people on top of this, from which we selected our 10 summit climbers. It's not the most glamorous endeavor. So we planned this expedition to within an inch of its life. This is a spreadsheet that I had prepared in January 2007. We didn't leave for Nepal till March. That gives the disposition of every single person and every group on the expedition across the whole 3-month period. And we pretty much followed this. So the yellow bars here are trek of group volunteers. The blue clusters in the middle are different groups of climbers, support climbers, the 2 film crews, and other logistical support. We shipped 26 metric tons of equipment out from the United Kingdom to Nepal, including nearly 400 of these blue plastic barrels, a huge number of cylinders of both oxygen for safety, but also calibration gases for the various devices, and all sorts of different challenges. For example, so our power requirements, for example, this was our generator cluster in Kathmandu getting serviced and set up to be sent up to the different labs. And this is the team. I'm not even going to tell you which person I am on there, but everybody's looking a lot younger than they are now. It's 18 years ago since we went to Everest. This is the investigator team in Kathmandu before we started. And we had 2 groups of participants. We had a trekker group who ascended to Everest Base Camp slightly more quickly than an investigator group. Of the investigator group, and the trekkers they go up and down in 3 weeks. The investigators all stayed at Everest Base Camp for up to 75 days, and a subset of them ascended high on Everest, some of them right up to the summit. We never lumped these together. So part of the rigor of the experiment was the fact that the oxygen or the hypoxic exposure was consistent and controlled. So the fact that the trekkers went up in 12 days means that from our perspective, they're not comparable with the investigators who go up in 15, but the comparisons between them can be very illustrative. This is the environment in which we're working. You can see Everest at the back, just under where it says, actually, no, just across. It's in the middle of that black bar. I'll try and quickly click OK, but I've got to work out where my mouse is. Oh, there we go. Apologies for the black bar. So Everest in the middle of the background, Namche Bazaar right down the bottom there, which is the Sherpa capital at about 3,500 meters. And you can see the path, the trail leading up, zigzagging up, which finally takes you about a week later up to Everest Base Camp. Oh, I'm not sure clicking was a good idea. Okay. That's better. This is the ascent profile of the two groups. So in the dashed line, the trekkers, and the continuous line, the base camp team. And then you can see as you look across expedition days on the horizontal axis, height and altitude on the vertical axis, the ascent profile of the climbers, including those who went to the summit. And these are the places. So a blue dot is somewhere we stayed overnight. A red dot was a fixed lab. So we had fixed labs. We tested people in London. We tested people in Kathmandu at about 1,400 meters, and then at Namche Bazaar, Foege, Everest Base Camp. And then you can see we've got two more higher on the mountain. Getting to Kathmandu is straightforward, big normal commercial jets. Getting above Kathmandu is a bit less straightforward. So you're in twin prop light aircraft that land on a very vulnerable strip high in the mountains at a place called Lukla, 2,800 meters. Well known for being the most dangerous, i.e. brackets, the most frequent accidents an airstrip in the world. And this is the airstrip. If you look at the far end, there's basically a cliff off the far end, and the person who's taken the picture, there's a cliff going upwards behind them at this end. So it's sort of perched on a little shelf on the edge of a valley. Above Lukla, the only way to move things, there's no motorized transport. It's either people carrying or animals carrying, typically yaks. And you can see here some yaks going across a bridge. This is low down where it's relatively warm and green, and you ascend over 10, 12 days to high up. This is the glacial moraine just below Everest Base Camp. You can see here, this is one of our exercise bikes going up on the back of a yak. Looking a little bit vulnerable. And in order to, again, ensure the reliability of measurements, we had a team devoted to metabolic and bike calibration for the exercise testing kit. So a couple of people went up and down, ensuring that our measurements were calibrated. And this is base camp. In the foreground, you can see a couple of laboratory tents. In the background, the medical and logistic tent. And then behind that, the Khumbu Icefall leading up into the Western Cwm. And the Khumbu Icefall is, as you probably know, a continuously moving river of ice. It's moving very slowly, but it is a dynamic entity. Inside those lab tents, we essentially got any measurement that we could do at sea level. So we're on, we use different voltage, but we're on 240 AC running blood gas machines, exercise testing, cardiac output monitors, near infrared spectroscopy, a whole host of different things, including some relatively invasive studies with arterial lines, nasogastric tubes. This is one of my least favorite studies. This was the two arterial lines, because if you monitor, if you use one of the less invasive cardiac output approaches using an arterial line, if you start taking samples, you lose your signal. So you need a second line for sampling, whilst you've got one for measurement. And this again gives you a feel of the attention we gave to making sure the measurements were accurate. So these are blood gases taken every 15 seconds to calibrate the gas exchange measured lactate threshold. So this is just making sure that what the exercise testing devices are telling us is accurate. So you can infer lactate from gas exchange, but we wanted to make sure that actually the measured lactate corresponded to the inferred lactate. And I think you'll be relieved to know I'm not going to share most of the data that we gathered. We've been here quite a long time. So all this information in the trekkers, and then in addition in the investigators, a whole host of other data points, with the consequence that we've got an enormous data set still from Everest. So about 13 person years of days of physiological data, more than 250,000 data points, nearly 2,000 exercise tests, more than 10,000 blood samples, which we've progressively worked through. And I'll show some of it to you now. So this is oxygen content data. It shows a fall off. The darkest bars are oxygen saturations. The grey bars are hemoglobin, and the white bars are oxygen content. And it's consistent across the next few slides. So oxygen saturations, as you ascend higher and higher, fall in the blood. Hemoglobin over time increases, and as a consequence, there's a change in oxygen content. Now you can see here the trekkers who are ascending over 12 days have a somewhat different profile from the investigators, so who are ascending over 15 days. So we're looking at in a little more detail. The trekkers, because they're going up a bit quicker, their oxygen content when they reach Everest Base Camp is around 13% down. So they're not quite maintaining sea level values. If you then measure their exercise capacity, this is progressively going London through higher and higher altitudes to Everest Base Camp at the right-hand end. The exercise capacity has fallen by about 35% by the time you reach Everest Base Camp. Keep that figure in mind. If you look at the investigators, they spend longer getting to Everest Base Camp, so their oxygen content when they get there is the same as it was at sea level. Their hemoglobins come up more. By the time they leave Everest Base Camp, it's up 18%. Their hemoglobin's gone up so much. Take the same measurements, say progressively going from London to the Western Cwm and actually higher. Turns out their fall in capacity to consume oxygen is 35% when they arrive at base camp with a oxygen content equivalent to sea level, and when they leave base camp with an oxygen content 18% higher than sea level. So your capacity to consume oxygen at high altitude is not related to the amount of oxygen you have in the blood. It's counterintuitive, but there's something else going on, which is almost certainly metabolic. If you look at, we did the highest in altitude exercise test ever conducted. There'd been a previous one about 50 years earlier on the Makalu Col with John West's team at about 7,500 meters. So we measured just under 8,000 meters. And at that height, the oxygen, measured peak oxygen consumption is down by about 60%. And if you bear in mind that there's resting oxygen consumption, so of the 40% you have left, all of us now are consuming probably 250 to 300 mils a minute of oxygen as we sit, just to keep the show on the road, to keep alive. So it's not that you've got 40% of your function left, you've maybe got 20% on top. And this is only the South Col. So these individuals have got nearly 1,000 meters to go before they reach the summit, which gives you an idea of why it's so hard to reach the summit without additional oxygen. This is really reinforcing this story that it's not all about the amount of oxygen in the blood. So this is oxygen consumption versus oxygen delivery. So on the vertical axis, it's actually oxygen content, but so you've got oxygen content on the vertical axis, which you would expect to be related to how much oxygen you can then consume. They're clearly completely unrelated. There is no relationship at all. It's a complete scattergram between your oxygen content change and your oxygen delivery change. This is, for those of you who are less fit, this is really encouraging. I love this data. Again, counterintuitively, the fitter you are, the greater amount you lose of your capacity to consume oxygen. And that's not just in absolute terms, because you'd expect if you can consume 5 liters a minute, you might lose a liter compared with, you might lose more than someone who can only consume 2 liters a minute. In relative terms, the fittest people lose the greatest amount. And that completely reflects what, if you talk to guides on Everest, they recognize this. They describe 2 phenotypes, which are typically American. I think that's because the majority of paying clients on Everest are American. And they describe the racing snake, so typical stereotype, New York hedge fund manager, skinny, been working out on his stair climber in the office every day, turns up at base camp, raring to go, really struggles, often doesn't get up. The other stereotype is the Midwest builder, smokes a bit, drinks a bit, didn't have time to train, potters up to base camp, potters up the mountain, gets to the top, potters down again and can't work out what all the fuss is about. And they literally describe those stereotypes and they completely fit this data. So what is going on? Well, we looked at the microcirculation, and if these work, which I think they will, if I can just get my cursor in the right place. There we go. So this is sea level versus high altitude for sublingual microcirculation. All you can see here is red blood cells. It's orthogonal polarized light microscopy. Here we go. Get that one going again. There we go. The left side is normal. The right side, the really sluggish flow, is what your sublingual microcirculation looks like at Everest Base Camp in a lowlander. So substantially deranged microcirculation with almost certainly deranged autoregulation going on. And if we look at, if I can advance, that if we look at the extreme end here where the 2 stars are for significance, the microcirculation of climbers who've been to the top of Everest was different from the microcirculation of individuals who'd stayed at base camp. So there is something phenotypically going on in terms of your microcirculatory blood flow separate from what's going on in the macrocirculation. And we started in 2007 to see some metabolic changes. So this is functional magnetic resonance imaging on the left, actually echo of diastolic index measures, E to A ratio and peak filling rates on the right. Measurements taken before, immediately after, and 6 months after the expedition. And the executive summary of this is there is a metabolic decrement in function and a measurable change in diastolic function of the heart in individuals who've returned from a trek to base camp. I'm really glad we did the 6-month follow-up data because we were starting to feel a little bit guilty when we saw the immediate follow-up data. It's completely recoverable. So this is essentially an inducible, reversible model of diastolic heart failure, simply as a result of acclimatizing to the altitude of Everest Base Camp. In order to get a really good measure of what's going on, you actually need tissue. So we took muscle biopsies. The image at the top is one of my favorite images from the whole expedition. It's not the most glamorous, but so the lady sitting down taking the muscle biopsy is a professor of anesthesia, and the chap from whom the biopsy is being taken is a professor of vascular surgery. I'll leave that with you for a moment. These are some of the muscle samples, and we basically showed what others had shown before, some morphological changes in muscle, changes in where the mitochondria are situated in the muscle, changes in mitochondrial breakdown. More importantly, when we did biochemical analysis of the muscles, we showed changes that suggested fundamental differences in the mitochondrial physiology. So the dark bars are after acclimatization, the white bars before, and you see changes in the levels of complex 1 and complex 4 in the mitochondria, and you see changes in mitochondrial biogenesis factors and metabolic factors, so PGC1 alpha and PAR alpha, suggesting that there are changes, but not proving that there are changes in mitochondrial function. We couldn't help ourselves, and we went back because we had a couple of residual questions. So in 2013, we didn't climb again, but we went back to base camp, another big cohort, to address 2 principal questions. One was, what about the Sherpas? Which we'd wanted to look at the first time, but there were significant concerns about the ethics of studying Sherpas who, it was said, may not understand what we were doing. We had so many Sherpas helping us in 2007 that there was an acceptance that actually they completely understood what we were up to, and it was perfectly reasonable for them to volunteer. So we studied a large Sherpa cohort in 2013, and we also were able to use techniques that were previously not available. So the microcirculatory imaging was of a higher quality, and you can see here a lowlander and a Sherpa at high altitude. The lowlander, not as bad as the previous one I showed you, but microcirculation is not as perky as it ought to be. Sherpa unchanged. There is no difference in a Sherpa microcirculation at base camp and back down in Kathmandu. So something that may explain their apparent phenotype of better performance. And I won't go through this in detail, but this is live, this is fresh muscle cellular respirometry. So using a device called an Ouroboros in situ at base camp on skeletal muscle, biopsied at base camp, taken straight out and put them straight into a respirometer. And this is a variety of measures of mitochondrial function, which in summary basically demonstrate that Sherpas have a lower level of mitochondrial leak and a higher level of mitochondrial efficiency than than lowlanders at base camp, which fits with the phenotypic differences you see. They are able to perform better at high altitude. So above base camp, you can see the Khumbu icefall there. Camp 1 and Camp 3 are overnight stops. Camp 2 and Camp 4, we had fixed laboratories, and then the balconies highlighted there for reasons that will become clear. This is the icefall. To give you a sense of scale, there's 2 climbers tucked away just up in the sort of middle left of center. And what they are on is, if you look above and to the right of them, there's a big cliff. What they're walking over is what one of those cliffs look like when they fall over. So literally, as you go day by day and week by week during the expedition, bits that were there at the beginning are no longer there at the end because of this continuously dynamic nature of glaciers. And the whole route is covered in fixed ropes on Everest. Because of the nature of the commercial climbing, there are ladders on the icefall and there are fixed ropes, which reduces some of the stress, but not all of it, because they're pretty creative in the way they use ladders. So this is a 3-ladder bridge, I think. We did have 4 ladder bridges. And this is what it looks like if you look down when you're walking across one of those, probably about 60, 70 feet to the bottom of the crevasse, which fascinated our camera teams. I can tell you this is the beginning of the expedition because they worked out about halfway through they could strap the cameras to their chest pointing downwards, and it was a lot easier than doing this business. This is so you can see the icefall sort of flattens out as you get to the top, and these are the crevasses at the top of the icefall. There are other hazards around, which we largely avoided, although we got some good pictures. And then this is Camp 2 up at about 6,400 meters. We're still on 240 volts AC. So we've got generators and solar panels up here and a fully functioning laboratory with exercise tests, mitochondrial, we didn't do mitochondria at this height, microcirculation, and a variety of other techniques, but with very different physiological measures. So this is the pulse oximeter in the middle there is at the start of an exercise test, before they've started exercising. So SATs in the mid 60s and a heart rate well over 100. And amongst other things, we're taking this is one of the places we took the arterial blood gases. Although we worked hard, we also had some fairly magic moments. So this is looking at the lab, the Camp 2 lab, and then looking across towards, actually you can see the shoulder of Choi Yu in the background. Above Camp 2, you have the Lhotse face with Camp 3 about halfway up on the way to the South Col. This is the Lhotse face, and the scale is massive. There is a campsite in the middle there with a significant number of tents, but you basically can't see them. It's about halfway up, just tucked on the way to the side. And there's a long sequence of fixed ropes. This is the relatively flat section at the bottom. Just as an aside, if you haven't seen this movie, it is a superb representation of what it's like to climb Everest. I don't know quite how they managed to do it, but it's remarkably accurate. It actually moved me to tears when I watched it on a flight. As you get higher up, it gets steeper and steeper. So this is coming up just below Camp 3. We're now in somewhat smaller tents. This is the camp at Camp 3. Views across towards Choi Yu is the big mountain in the middle, about 10 miles away. And then above Camp 3, big traverse across the so-called yellow band for obvious reasons, with the South Col behind. And at this point, we're climbing with additional oxygen for the safety factor, as the mortality rate on Everest is halved if you use oxygen than if you don't use oxygen. And as a medical research expedition, there was no decision really there. We clearly had to use oxygen, but all the measurements were taken after at least half an hour off oxygen. This is the lab on the South Col. We're down to 24 volt electricity, but still doing exercise tests. We had transcranial Doppler up here. We had the microcirculatory measurements, near infrared spectroscopy. And we had a climbing team of 15, of who 10 were on the summit team and 5 were there to assist with science and assist with the inevitable rescues. So we got involved in several rescues. And I mean, if you put doctors on the South Col, they're inevitably going to be called to help. And we had planned for the eventuality that we could help, but we would not stop. It wouldn't stop us conducting the science as well. When you climb Everest, you typically climb at night. So we set out at 10 o'clock at night and you head off for hours and hours until first light. So you're just climbing by the light of head torch. You can hear a bit of scratching in front, a bit of scratching behind from other people's crampons. About 5:30 in the morning, The sun starts to come up. I thought it was great. You could see where you're going. It got a little bit warmer. My very good friend Dan Martin, who I was climbing with, thought it was absolutely terrible. He said I could see the way down for the first time. It looked awful. We got to the south summit, which is where the bridge dips a bit after the south summit. So this is a picture from the south summit looking across at the Hillary Step. You can see highlighted there, and a couple of climbers on the Hillary Step. And then above the Hillary Step, remarkably, this is what the track looked like when we climbed. So we deliberately descended when the first groups went up in order to avoid the congestion that takes place. So we went up and had a very clear run at the summit. But by the time we went there, nearly 400 people had gone up and the weather had been good. So this is how the track looked on the summit ridge, the southeast ridge of Everest that year. I'm going to show you a short section of video with sound. And if you listen carefully to the sound, so you can hear, this is a Sherpa with a camera with sound recording on their helmet, and you can hear the ventilation, which gives you a feel for the level of exertion at this sort of altitude. This is before the south, just coming down from the south summit. And then above the Hillary Step. So this chap is an elite felt runner at sea. So that's the first proper look over to the Tibetan Plateau, having climbed from the Nepali side. We successfully got 8 of our climbing team, a couple of climbing cameramen and several Sherpas to the summit. But we didn't do the one piece of science that we wanted to do really high, for reasons I'll explain in a moment. This is the view from the summit. You can see Makali there, the 5th highest mountain in the foreground, and far away in the background, Kanchenjunga, which is about 150 miles away, the 3rd highest mountain on the Indian border. We'd planned to take arterial blood gases on the summit, and we'd done a lot of preparation to do that. But the weather, although pretty good, was still, we had about 20 knots of wind, and it was about minus 20 degrees. So in the end, we descended about 400 meters to a structure called the balcony, put up this little shelter and took 4 arterial samples, femoral arterial samples in there, along with a venous sample for comparison. And then the chap standing up, Pasang Sherpa, literally ran them down from here, 8,400 meters through the South Col, all the way down to the benchtop blood gas analyzer we had at Camp 2 in less than 2 hours. And he stopped for a short cup of tea on the South Col. So this is straightforward femoral stab, glass syringes, so there's no oxygen absorption in the syringes, and then a calibrated blood gas machine waiting for them when they arrived down at Camp 2. This is how we showed it. You've seen some of these images already in the New England Journal. I'm going to show you a slightly different set of images to close, which is the blood gas plotted as partial pressure of oxygen versus partial pressure of carbon dioxide. Sorry, they're in European numbers, but if you multiply by 7.5, you get millimeters of mercury. So these would be values typical of any of us now in this room. So arterial PaCO2 is about 5 kilopascals, so 45 millimeters of mercury, and the oxygen levels are sort of 12, 13, 14 kilopascals. Typically in intensive care or anesthesia, we start to get alarmed when the blood level of oxygen falls below about 60 millimeters of mercury, about 8 kilopascals. So we turn up the oxygen, we put people on ventilators. Everybody at Everest Base Camp is comfortably below that level. They're all feeling okay. They're not necessarily feeling perfect, but they're all feeling okay. They're all functioning normally, but they have an arterial blood gas that would generate considerable alarm in a clinical context. And as you get higher and higher, the oxygen partial pressure in the blood gets lower. Not surprisingly, there's less oxygen available, and the partial pressure of carbon dioxide gets lower because of the hypoxic drive to breathe. And there's actually a little bit of a relationship such that those who breathe the most manage to get the higher level of oxygen in the blood, but they also blow off more carbon dioxide. And that's true at 6,400 meters. It's true at 7,100 meters. And these are the highest in altitude measures of oxygen in the blood ever taken at 8,400 meters on the balcony. And here we have it in millimeters of mercury. You can see the 4 individual participants. All of these are functioning really normally. The 2 individuals, subject 1 and subject 2, were the 2 who took the blood gases. So we took 2 blood gases each from ourselves and then from the other participants. And you can see a range from just under 30 millimeters of mercury to just over 19 millimeters of mercury. In people who are talking fluently, they've actually walked to get to where they were. So presumably their oxygen tension was lower when they were exercising. They've had a bit of time to rest and get their oxygen back up again to 19 millimeters of mercury. And this sort of profound hypoxemia is incredibly rare. I mean, when we looked at it when we came back, there are a handful of clinical case reports, typically in cardiac arrest. There's some rather disturbing ones from the 70s of individuals with opioid overdose who were left on a trolley, a gurney in the emergency department, and then somebody came along and took a blood gas and discovered they were profoundly hypoxic, and they survived. The other context is in the animal kingdom. So Warren Zapol's data from diving Weddell seals shows profound hypoxemia when they surface of almost exactly the same level. And it's interesting, you would imagine it's not an evolutionarily stable strategy to get the surfacing decision wrong. So you would imagine they surface at the point when they're properly running out of oxygen, and their PaO2 is about 2.5 kilopascals, so just over, low 20s millimeters of mercury, so similar level. And there's one other context when this is completely normal, which every single one of us in this room has experienced, which is the fetus in utero. And it turns out, and I'm certainly not a neonatologist, but when you talk to neonatologists, 2.5 kilopascals is roughly the threshold between thriving and not thriving. So if you can keep your arterial oxygen tension as a fetus above that, organogenesis continues, normal development continues, and if you don't, you get failure to thrive in utero. So one could speculate that there is a flaw in what mammal physiology needs in terms of oxygen tension. I'm going to close there. I think we're running or have run out of time. It was a massive team effort. This is another one of my favorite photos. This is just after we descended from the summit up at Camp 2. And you can see the Sherpas and the climbers and the investigators all intermingled. And this really, for me, is a massive reminder of the fact we couldn't have done it without the Sherpas. They got a lot of the kit up high on the mountain, including onto the South Col. They helped us climb to the summit and were integral to the whole endeavor. This is 2013. So in 2007, I led the expedition. I summited the mountain. I think it was probably roughly equivalent as a challenge to take 3 children under the age of 3 to Kathmandu and run a lab with my wife there, as it was during the 2007 expedition. We actually have 2 boys and a girl, but the daughter insisted that they had a princess party, so the boys complied and put on dresses for the occasion. I'm going to leave it there. I really appreciate you listening, and I kind of hope that Tom, looking down on this, would have enjoyed that, because he certainly inspired some of it. So I'm incredibly grateful to Grocott and others for the opportunity to come here and give this lecture. Thank you.